Hi, everyone. Today I am very excited to introduce Dr. Scott Longing, who is an associate professor at Texas Tech University in Lubbock. His research lab focuses on endemic insects of the southern U.S. and the environmental conditions associated with occurrences, species-level biodiversity, and community structure of terrestrial and aquatic insect communities. An overall goal of the research that his lab does is to provide information to help conserve native biodiversity and ecological services and processes that the ecology supports. He is a collaborator on the Southern States Pollinator team, which is a collaborative multi-university project aimed at engaging citizen scientists to compile data on the of foraging insect pollinators on flowering plants in home and community gardens. He also participates in outreach and engagement with local and regional farmers interested in adopting conservation practices to benefit pollinators. As a part of this outreach, his lab has the facilities and equipment to establish pollinator habitats through direct sowing or drilling seed to enhance pollinator resources on working farms. He's published 23 articles and 2 book chapters. Today, he'll be talking about insect pollinator conservation in the Southern High Plains. Thank you, Scott. Take it away.
Thank you, Amanda, for the introduction and getting this series together. I'm excited to be participating and grateful that you invited me. Today, I'm going to be sharing some research. Actually, it's some research highlights. Meaning that I'm going to give kind of an overview of some of my research that's been that we've been working on in my lab. The topics that we'll cover that I'll go over, I'll give a little background on pollinators. I think we all know the issues surrounding pollinators and their decline and issues related to environmental stressors, especially in our region where there's a lot of intensified agriculture and related stressors. So we'll discuss that background a little bit related to the Southern High Plains. Within a USDA Conservation Innovation Grant, back in 2015, it was from 2015 to 2018. There were several studies that were conducted as part of that. So this is going to, in large part, discuss some of those outcomes of those studies within that SIG. So with that, I'd like to acknowledge NRCS for funding that project. It was a big learning curve for us, both in planting and also in learning the bee biodiversity, which is a pretty big task. My PhD student that just graduated, he's graduating this semester. He was the one that really dug into the bee taxonomy. He's more of a bee taxonomist than I am. I directed all of the research, but he really deserves credit for a lot of this work that was conducted, as well as other graduate students. I'll talk about several projects and different graduate students led those projects. So the value of pollination services is estimated at $212 billion globally. Over 80 different crops benefit from insect pollination to increase yield or increase seed set or seed cotton weight in the case of seed cotton weight in the case of cotton. Pollinators also help maintain flowering plant communities. They provide food and shelter for wildlife, and they also support plant reproduction that provides material for livestock. So we know that there is an essential component of what we do and what we use, and certainly from an economic perspective, they provide significant value to crop pollination. A note here I like to make, a note that's important to remember, and this was a paper that came out, I want to say a couple of years ago. I think some of you might know what that the paper I'm referring to here. I wish I would have cited it here, but anyway, the conservation of pollination ecosystem services might not be the same as the conservation of pollinator biodiversity. So that paper showed that within a crop, and my apologies, I forgot what crop it was, but they showed that a small fraction of the overall bee biodiversity locally was contributing to, the movement of pollen within that crop. So when we talk about the conservation of pollinator biodiversity and native bee biodiversity, that might not be the same as the conservation of pollination services, which might be a subset of the overall diversity that we know. And certainly honeybees are one of the best pollinators. So that one certainly falls into that. So in the Southern High Plains, we have experienced intensified agriculture. Pollinators are understudied overall in the region. We are continually surprised at the abundances and diversity of bees we're seeing, even in wheat cover crops, South Texas corn. We're finding lots of bees in those crops, and really don't have a clear understanding of why they're there or if they're using the resources in those habitats that we manage. So there are about 900, estimated to be about 900 native bee species in Texas. That's probably an under estimate. We know here in this 14 county region on the Southern High Plains, I'll talk about it later, but there's about 250, 260 documented native bees. We assume that's probably a underestimate, but across the state of Texas, about 900 bee species are known. So on the Southern High Plains, bees deal with quite a bit of change, right? Landscape change, from habitat fragmentation that creates non-contiguous resources spatially and temporally. So, with crop planting, harvesting, and management, there's mass emergences of flowers, usually with those crops, say in July for cotton. So that's a different sort of resource for native bees. Are they using that? Well, studies indicate that they are in cotton, and we've certainly gathered some data that show that they are in other types of these managed landscapes, but we just don't know exactly what they're doing there or what resources they might be using. But those are important questions to understand spatially and temporally how resources across a highly impacted region influences bee communities. A new area that we're kind of getting into here is exposure to environmental stressors, especially agrochemicals. So, this is a great region to understand what drives bee exposure to agrochemicals in terms of habitat fragmentation, resource use or degree of specialization, temporally, as far as farm management, how does that affect exposure, and also species traits or taxon traits. So things like body size and foraging distance, how does that influence how individual bees in the community are affected, and therefore might affect community structure or population conditions. Species overlap is another issue, and that's regarding both resources and where these bees physically overlap in terms of our honeybees and specialized bees foraging on the same plant, or honeybees being generalist. Are they going to go to a specialized plant and potentially affect or perhaps transport agrochemicals to that resource? So Coe et al. In 2015 identified our region as one of the, I believe it was 14 regions in the US that was listed as these places of concern regarding pollinator abundances. So these locations were modeled as having potential low abundances based on, I think in large part it was based on how much agriculture was in the area and how much land cover change had occurred in that area. So of course, all the agricultural regions were highlighted in that, and because, you know, it concurrent with, you know, these environmental stressors is the need simply for mini crops, pollination services. So, you know, these agricultural regions are both places that are suggested to be low in pollinators, but also places where, you know, we need those pollinators, especially for some of the specialty crops that require insect pollinators. Okay, so I'm going to go into the USDA SIG project. And again, within this project, there were several different studies that we conducted. I'm going to hit the highlights. I'm going to show you a lot of results. I won't show you a lot of methods, but if there's anything in here that you want to discuss, you can certainly contact me if you want any references or methods further than what I'll explain here. I wanted to pack more results in than explaining methods to you for any extended time. So briefly, these are our sites for our SIG project that was conducted. There were 42 different farmland habitats. So we described those could be pivot corners or bar ditches, fields that were either in CRP or were just left unmanaged. We had some vegetable farms, orchards. We kind of did a shotgun approach. We realized that we weren't, you know, we weren't replicating these farm types, but we did want to get representative of all different types of farms. So we did that. We kind of picked a bunch of different types just to be on these and kind of see what the bee communities were at those locations. One thing we did have was a fairly decent soil gradient from sort of a loamy clay to more sand, from northwest to northeast to southwest gradient. So we were able to determine if soil might play a role in bee communities occurring across this region. As the project got going, we realized that you know, there were a lot of local farmers market type farms that were interested in this project. A lot of the larger farms, they were certainly cooperative, and we enjoyed working with them. It was just their scale of operation is much bigger, so you run into some you know, problems that are just associated with operation and scale because they're a little they have a little more acreage to manage and so forth. So the local farmers market farms, they kind of came on a little bit later in the project, but we were able to fit them in. And at some of those locations, we did have some pretty successful plantings. So at a subset of these farms, we planted a wildflower mix using this Truax seed drill here, this little 40-horse tractor. We used the Native American seed Be Happy mix quite a bit. That was one that was in this field pictured here. My graduate student, Chris, he planted that, and it came up very successfully. Gaillardia was a big hit. We have found that in these plantings, you know, it's very rainfall or irrigation dependent. So basically where you have any sort of irrigation, you know, you'll get germination and you'll get some floral abundance and floral diversity. Where you don't have irrigation, you really have to rely on the subsequent year prep, and you kind of have to get the seeds down on clean soil. And you know, it's a it's a fine line between, when the harvester ants will carry all your seed away, and when, and then the rains, because we get most of the rain in September. So it's a little bit tricky there to get seed down, but you want that good winter rain on these seeds. If you don't get that, which in I think 2017 was a dry year, if I remember right, which was in the middle of our project. You get pretty low germination. So we saw that across the board. This 30 species mix, if we kind of let it do its own thing, then 5 or 6 species a year in terms of the flowering diversity, that's about what we would see.
There's a question in the chat. Do you want me to ask it now? Is that okay?
Yeah.
Yeah. So someone has asked, has the use of cover crops and crop rotation helped the bee pollinator populations, like the use of more diverse crops? Has that been addressed? In the pictures, there's a lot of bare ground.
Yeah, so Chris is planting there. That was, I believe that was we planted some native grasses in that field in June, and then planted over in October, planted the forbs. Yeah, there's a lot of bare ground there. We were trying to establish a native prairie, which we were successful at. We had 5 species of native grasses, and then the Bee Happy mix in that field. Cover crops are certainly beneficial in many ways. And I think their benefit is not maybe not as known as it should be, because the early season resources for pollinators in this region is very important because of the amount of land, like you see here, that just is getting prepped, right, for planting. So, what percent is bare ground in March? We haven't determined that, but I would guess it would be a pretty high percentage. So from my opinion, any cover crop that could be used in the winter that could bloom early in the season, or a winter crop would be, a non-cover crop would be very advantageous for pollinators. Where I have a student right now that's has been since early March putting bee bowls out and sampling other ways in wheat and alfalfa. And again, we're finding even in March, the first date was about March 5th. The bees were a little bit low then, but the next sampling date, which was about the second week of March, it was, the cups were full of bees. So we're going through those now from those cover crops. So that's a good question. Cover crops are very important and need to be, I think need to be used a little more regarding, pollinator habitat enhancement and resources. Okay, so here's the method slide. This is going to be fairly quick here. At each of those dots on the previous map, we set up transects. So we set up 2 meters by 60 meter long transects. We had a linear transect, and then we had what's called a random walk transect. So we did a 60 meter transect, and then we did a random walk transect where you walk, I think it was 15 or 30 meters. You take a left or right direction, and you do that again and kind of randomly walk through there. That's to hit, blooms. Often if you stratify sampling in these habitats and you don't do hunt sampling, you'll miss blooms because they're simply not very dense. So we also did what's called hunt sampling. So at all the locations when things were blooming, we would go to that bloom and collect bees at that bloom. We combined all the bees for the community data set. We combined that across all methods. From that community, we developed some state variables, generic richness, abundance, and then a few metrics, diversity metrics, so evenness, Shannon's index, and then Simpson's diversity index we calculated from the community data. And then we used generalized mixed modeling and ordination analysis to look at relationships of local and landscape level habitats across both years on those community state variables. And this was concurrent with plantings to, to enhance and restore pollinator habitat at these farms. We initially had a nice before-after control impact type design set up to where we could take these 42 sites, at some of them restore habitat and use that seed drill and plant, and then assess changes there, and then not plant at the other locations where we had been sampling to see if there's changes, right, at that place, at the before and after those flowers were planted. However, we didn't get great success at the locations where we did get good germination and good stand growth. It was nice, and I regret I don't have more photos to put in here, but we did have some good growth, particularly at some of the farmers market farms. Onion Shed Farms is one where we had a nice stand of Gaillardia surrounding a hoop house that was put up, and again, they grew there because the farmer was growing blackberries. So, and actually what happened was we seeded his entire place. We drilled his entire, their entire farm. After we did that, a month later, they put in the blackberries and the irrigation lines at the bottom of the blackberry vines and turned the water on. So it was this beautiful blackberry arrangement with a lot of Gaillardia and Coreopsis and bee balm and things coming up. But again, it was because that irrigation water was available from those blackberries. So at each site, this is some of the data that we collected. So the site number, the site name. So this Bingham farm, this was in Terry County. So within that farm, they had what we called wildland, which was a little strip of canyon land that ran through the farm. They also had organic grapes, and they had organic cotton at this farm. In Lubbock, we had a couple of vegetable farms, Apple Country Orchard, South Plains Food Bank Garden. So all different types of habitats, really, to collect bees and measure these habitat conditions. So the size, hectares of the location where we collected bees, what type of farm, whether it had a CRP or not, or whether it had CRP plantings or not, and whether it had a Playa on the property or not. Soil series, and then the dates we sampled. So we sampled on 4 occasions, 3 in 2016, and then 1 in 2017. So in short, these are the results of the GLM, the generalized mixed models, showing the year sampled, the season. So the season, then our total 2016. So that was the combined data, and then the May, June of the 2017. So the effect here, CRP, farm, Playa, and then soil. So we see over here in bold where we have significance. So for floral diversity, April and May season, CRP had an effect on floral diversity. In this case, floral Playa had an effect on floral diversity. And we did the same thing for bees. So we had bee abundance, bee richness, and then Shannon's diversity. And I had that table in here. I thought I had changed that table with this floral richness and abundance table because that was less interesting to me. But I believe on the next slide, we do have some results of bee richness, bee abundance, and bee diversity across landscape structure. Okay, so this is, this was an error on my part. I regret that. This was this table here was supposed to be floral or bee abundance, bee richness, and then the Shannon's index. And we did see with that bee abundance and generic richness data that there were effects of CRP and Playa in 2016, but not 2017. So for those 2016 3 sampling events, the combined data, we did see effects of CRP and Playa, but not farm and not soil type. But for the 2017 date, we didn't see that. So, and again, this is the floral richness and abundance across those different factors. Okay,
Sorry to interrupt. This is Susanna. Just somebody had a question about the methods you used. I think we have an audience that's not as familiar with the terms.
Okay.
So they specifically asked about netting and bee bowls and transects.
Okay. Okay, so bee bowls are, I should have put a photo of that in here. Bee bowls are 3.5 ounce plastic solo cups. There's really nothing to them. They're painted fluorescent yellow or fluorescent blue or white, and the bees are attracted to those colors. We put a solution of water and liquid detergent in the bee bowls. So that's to charge them, and that's how you collect the bees. The bees will be attracted to the color and they'll fly into the bowl. The detergent breaks the surface tension of the water and the bees essentially drown. And we go back after, we do it a couple of ways. We either go back after 4 hours. So if we go to locations where we set up a big trap line of bee bowls, then we will set them out and then go back and get them the same day. So we don't have to go back the next day. And we still find a lot of bees with 4-hour samples. But we usually set out the bee bowls for 24 hours. Transect methods, that is just the 20 by, or the 60 by 2 meter transect that we use to hand net. That's called a belt transect, where you just delineate a length and a width of the transect and you collect everything that's down through there. And then hand netting, that was using the hunt sampling, where we go to the site and we locate, you know, flowers that are blooming at that time, and we collect off of those.
Thank you. We had one more quick question. What is
Yes.
Playa and what is CRP?
Okay. A playa is that's kind of our little, well, not little, they're very numerous, but those are our aquatic habitats here on the high plains. Those are the watersheds, so to speak. Each little playa is a, you know, the high plains is very flat over here in West Texas. So, you know, we don't have big steep valleys and things like that, but we do have very gradual landscapes. So water kind of pools up at areas, you know, depending on the terrain, and where that occurs, we have these shallow depressions in the terrain that hold water, and they might hold water ephemerally. So, you know, here one day and then it dries up fairly quickly, or they might be more perennial habitat, depending on the soil, where they hold water a little longer. So a playa is just basically a pond on the high plains, except we have about 23,000 playas. And CRP stands for Conservation Reserve Program. That's part of the USDA CRP program, where farmers can elect into the program and plant habitat for wildlife and use different methods to enhance wildlife on farms.
Thank you.
Okay, so another landscape analysis, and this is the only habitat type analyses that I'll show you. We'll move on after this, but another landscape level analysis that we conducted, we delineated buffers around each of our collecting sites. So we delineated a 200-meter buffer, a 500-meter buffer, and a 1,000-meter buffer. Within those buffers, we calculated the percent land cover, and basically just wanted to look at what sort of land cover composition and patch richness, so the patchiness of habitats, You know, whether around this sampling location, whether it's entirely cotton, you know, 80% cotton, or does that land surrounding that bee sampling location, does it have more, a stronger mix, more heterogeneity in that land cover? So is there more grassland or other land, which is this barren land that we've been seeing? So we did see some effects, but it was dependent on scale. So at the 200 meter scale, we saw a relationship between bee richness and developed land, between grassland and bee richness, and between other, which was the barren land, and bee richness. Overall, we didn't see a lot of significant effects. Significant factors here jumping out at explaining these bee communities. And one reason we think is because there's so much landscape heterogeneity, and our sampling design didn't replicate, you know, landscape heterogeneity. We didn't select, you know, X number of sites that had high heterogeneity versus low. We simply selected based on farm type, and then whatever landscape surrounded that farm, that was what it was. And we actually calculated that overall, on average, you know, every spot we clipped out generally had about 30% grassland habitat. So meaning that you know, over this area, that might be close to what it would be if you were to kind of look at multiple scales or look at the region as a whole. Okay, so on to some other stuff. That was just relationships of habitat, and again, overall, we didn't see a lot of relationships between local habitat and landscape habitat, and that's again a big reason was because there's as the photo showed you at the beginning here, it was a big it's a big mix of landscape types, right? Unless you replicate different landscape types, your effect size is likely going to be low with that. So
could you take just a second to explain abundance, richness, and diversity?
Sure. Okay, so bee richness, that is simply the number of taxa, and that should probably say generic richness, because we took our bees to the, for this analysis, we took them to the level of genus. So that's generic richness. So that's just the number of bee genera. Bee abundance, that's the total abundance for that sampling period. So we had the 4 different sampling periods for each site. So that's the total number of individual bees. And then bee diversity, that's the Shannon's index. That takes it into account both the abundance and the frequency or the occurrences of those species within the data. So it's looking at both the counts of taxa and then the frequency of that taxa. And that's the Shannon's index. Okay, so we put together another part of this work was simply to understand more about what the bee biodiversity was here in the High Plains. Not that we were going to be the sole doers of that, because there are other studies going on in the region that are kind of looking at the same thing. But we, coincidentally, over the last 3 or 4 years, there have been a succession of studies that have been conducted in the High Plains that have really given us some good background data to build these species lists. So we developed a checklist. This was part of Samuel's dissertation research. So across a 14 county region, so these are the 41 counties. So we did treat the entire High Plains region, but for this checklist, we looked at a 14 county region. In this region, these are the studies that are that have sampled bee communities and identified bees in this region to species. So if you're interested in bee research, especially bee biodiversity, these are kind of the studies that have put some of the information together. There are probably more. There's some that predate 93, but these are some of the more contemporary studies here. So Maldonado found 15 species in hybrid sunflower. The Berger study was really the biggest assessment of bees in the region prior to these more recent studies. So the Berger study found 67 bee genera. They were looking at cotton, alfalfa, wildflowers, and some other types of plants. Alicia Patridge, an MS student here, she conducted an urban bee study and found 20 bee genera in urban habitats. Angie Bigosh was a PhD student.
Scott, I think you might have frozen.
Excuse me?
Oh, I said, I think your screen froze with your video. So if you need to, feel free to turn the video part off, and that might help with the broadband.
Okay. Alrighty. Okay, so Bigosh found 127 species. So that was a big study there that really helped with what we know about the biodiversity of bees here in the region. 58 genera. Auerbach et al. 2019. That paper was produced from a bee data set that came from the Muleshoe National Wildlife Refuge. So over 2 years there was extensive collecting. They were looking at prairie dog towns and differences in bee communities between prairie dog town and non-prairie dog town habitat. In conducting that study, they really put together a big list of bee species, 163 species and 41 genera. And then Disqua 2021, from this study, from the SIG study, 106 morpho species and 49 genera. So what we did for this checklist, we mined data from certainly all the available published articles. And we also looked at several collections around the state and regionally, and we downloaded data related to the Anthophila, the bees, and we assessed that data and put together some information regarding the bees occurring in the region. So this came from museum records, published studies, and recent surveys. This checklist is for 14 counties in the Southern High Plains. So overall, from those sources that I just mentioned on the previous slide, 286 bee species collected and reported to date. So that's the number we came up with after looking at all of these different sources and all of the different published articles. So 286 bee species occurring in this 14 county region. That's an increase of 158 species from the 128 that were previously reported. So that's these new studies, they have really added to what we know about the bee biodiversity in this region. So recent studies, including ours, have documented about 60% of the historical species reported, 51 species previously reported, but not documented in recent surveys. The most common bee species based on the number of counties of occurrence and the total number of specimens. So 30 to 40% of all specimens reported to date for this 14 county region are these three species. Agapostemon angelicus, Melissodes communis, and Melissodes coreopsis. Three very common species occurring in this region. 10 species were reported for all 14 counties. 92 specimens reported from a single county. So this is where we can really increase the county records here, simply because in some counties, Andrews County, for example, down south, there's just simply not much known from those different counties. 25 species of bees occurred as singletons. That means that for those 25 species, there was only one specimen that was reported, whether that was from the museum record or from those articles that dealt with surveying bees. 3 species have not been recorded for over 80 years. Nomata autumnalis, Melisodes microstricta, and Andrena illinoisensis. These species have not been collected in or reported, excuse me, from any of those papers that were listed previously. Several species are known in the region, but are collected very sparsely, even when they might be in high abundance on specialized plants, simply because the sampling devices might not be effective at collecting, specialized bees. This is a family level assessment of what we know in the region based on this analysis. So Apidae, 30 genera, 110 species. Andrenidae, 7 genera and 61 species. Megachilidae, 12 genera, we'll see some of those, and 51 species. Halictidae, 8 genera and 47 species. And then the coletids, there are 2 genera and 17 species. Okay, so so I just talked about specialization a little bit there, and the fact that some of these data sets we're finding there's sparse occurrences of some of these specialized taxa that we're starting to learn a little bit more about. One example of a specialized bee that we've certainly focused on recently, but typical community level studies sort of leave these specialized bees out. And that might be because of the sampling method. You know, for many of these bees, if you're not going to the exact plant at a specific time, our conventional methods of collecting bees might not work. And it's known that there are diverse specialist bees. So this is an example of one, Sphacotisoma prati. It's a little Rhophytini bee. It's a specialist on Nama hispidum. Nama hispidum is also called bristly Nama or purple mat. So we're interested in understanding what are the effects of habitat fragmentation and disturbance and agrochemicals on specialized bees and other bees, but especially these that depend on certain types of plants. And in the case of this bee, this is the only pollen that it will collect. It'll collect pollen from Nama, and then that's what it feeds to its brood, and it won't forage on too many other things if Nama's there in the region or in the locale. So this is Nama hispidum. This is a specialized plant for this bee that's in this corolla here of this flower. This is Sphacotisoma prati. These are 2 females. You can tell that they're females by the pollen hairs on the hind legs. So those are used to collect the pollen, take the pollen back to the soil and feed it to the brood, mass provision the pollen, lay an egg on the pollen, and then the larva hatches, eats the pollen, and emerges out of the soil. The distribution of Nama hispidum is from Southern California down into northern Mexico, and then over into southeastern New Mexico, central Texas. The bee overlaps with the plant. So Sphacotisoma prati, its distribution essentially mirrors this distribution of Nama hispidum. So it's a specialist on this plant. And there are other bees that we're finding that are specialists on this plant, and there are other known small bees of this size that are specialists on this plant. The diameter of this corolla here this flower is about 7 millimeters. So this is a very tiny bee, although it fits perfectly, down in this this native plant. This is a very, very small bee here. On the left is the female, so you can see the pollen collecting hairs. On the right is the male, a little bit different shaped head, longer antenna, and then you don't see the pollen collecting structures on the hind legs. So all this male wants to do is fly around this plant. And if you ever get a chance to see this plant, it's worth watching. Midsummer when there's a lot of blooms on it, you'll see these bees. This is the female trying to find pollen. The males fly at full speed and they brush the females. We don't know exactly what they're doing, but they'll fly and they'll kind of hit the female and they'll either bounce off or they'll, if mating occurs, then they'll ball up on the flower in just a split second and then take back off. So it's kind of a fun system to watch. A lot of action on it, a lot of blooms. These specialist bees, they not only forage on this plant, only, but they also nest right below the plant. So this is a entrance to a nest of Sphaconosoma prati, and this is the flower. You can see that the corolla opening is about the same diameter of the nesting of the nest here. We also find that on many occasions, these bees will kind of use these dried cracks in the soil. We see that a little bit. We don't know if those cracks are facilitating nesting or what's going on there, but we find holes often along these little fissures in the soil. This little spider here, we have actually been seeing these spiders on more than this occasion here. We don't know what this little thing's doing, but perhaps it's a sit and wait predator and it's waiting on these bees to emerge. That would be a great strategy because these bees, they're newly emerged, so some haven't been out of the soil yet. So, that would be a bad fate to come face to face with the spider as you leave your nesting ground, but we think that's might be what's happening. This plant, whoops. So these are photos actually taken yesterday. We went over to our farm. We're looking at some plots to grow some sesame and look at sesame hybridization and the presence of honeybees and other bees at this location. But this was a growing patch of Nama on the edge of the cotton field. So here's the prepped cotton field. If you see these light areas here, these are the Nama patches. There's one there, there's a fairly big one there, and there's a fairly decent sized patch there. But anyway, the plant produces thousands, if not millions, of very tiny seeds. So we're trying to understand how farming practices influence this specialized plant. And in some cases, it actually might be promoting this plant. The plant doesn't like competition. The seeds are really small. We think the seeds get buried easily. We think tilling might promote those seeds coming to the top of the soil. And when habitats are protected, so this is newly disturbed soil. This weather station here, the boom had to be turned before it hit this weather station, so it left this area that was relatively undisturbed compared to this tilled soil right here. Of course, this was disturbed not too long ago, brought those Nama seeds up to the top, they germinated, and this is going to be a big patch of purple mat that's going to attract native bees, likely right to this location. And those bees will in turn likely nest right in this area. So we're trying to understand the interplay of disturbance and farming practices and where these specialized plants, where they spring up. Historically, it would have been because of maybe bison trampling or fire, other soil disturbances that might expose some of these seeds. So we're trying to learn a little bit about its preference for the locations where it germinates and forms some high floral abundances for these native bees. Okay, we'll pivot, but kind of stay on the same pollinator theme here. This is another emerging area of research that we're getting into. We're trying to determine ultimately how habitat fragmentation and species traits and resource use, whether the bee is specialized or not, as in the case of Scotostoma prati, how do those conditions influence whether a bee is exposed to agrochemicals or not within this landscape? And, you know, in learning, knowing more about that, you know, we might be able to prescribe better conditions for conservation of perhaps specialized species, or perhaps prescribe ways to, you know, keep honeybee foraging within a certain location or within a certain resource. So understanding how species traits, resource use, habitat fragmentation, how all those influence agrochemical exposure is key. This paper looked at, we collected 200 or 382 bees, and we, at each location, we combined them by genus. So from each location, we had a genus sample. Each of those sample of bees were analyzed for 3 neonicotinoid compounds. Neonicotinoids are the primary insecticide used to, or they're one of the primary insecticides used to coat cotton seeds and other crop seeds, so that when that seed germinates, that insecticide is systemically taken up in the plant to provide some pest control. So the neonicotinoids are a big group of insecticides that are broadly used across the region. So we simply wanted to ask, during the time of cotton planting, if we collect 382 bees, how many of these bees at the genus level, because we wanted to capture those species traits that were associated with that genus, you know, what's the frequency of exposure? So we found 20 of 382 bees had detectable levels of one of the 3 neonicotinoids, okay? And Apis was the most frequently, was the genus with the highest number of positive detections. It turns out that Apis was also one of the largest bees, along with Melisodes. This relationship showed a marginally significant trend at the alpha level of 0.1. It wasn't a strong relationship, but overall it shows this emerging trend that larger bees that forage further might be more exposed to agrochemicals, which makes sense, right? If a bee is going to fly further, visit more plants, it's likely going to be more exposed to agrochemicals. However, we did find that one of our bees, Perdita, it's a very small bee, just a few millimeters. It had the highest concentrations, the highest 3 concentrations of neonicotinoids. We have seen Perdita forage. We've seen it on silverleaf nightshade next to fields. So it is one that will use some of these invasive type plants. So maybe that's leading to these high incidences of agrochemical neonicotinoid concentrations. But overall, this analysis showed us that it might be in part species traits that drive exposure, but within that, those conditions of resource use and specialization. So are honeybees, if they forage on Nama, which we know they do, are they vectoring agrochemicals to that location? So on that same note, we conducted a cavity nesting bee study. We wanted to ask, because trap nests are very conducive for setting out. They're essentially passive sampling devices. The bees colonize them, you can collect the contents, and you can analyze what's in the contents. So we conducted 2 separate studies. They were both, one was conducted by Caleb Tomlinson and one was conducted by Eric Peterson. These were 2 master's students that have recently completed their work. For Caleb's study, we looked at the differences between a drilled nest. So this is the bottom right corner down here. This is the sort of the homemade drilled nest. We got a 4x4 post. We drilled different sized holes. So 5/32, 7/32, 5/16, and then 1/8. We had different sized holes for these sort of DIY trap nests here. And then we bought the Crown, I forgot the name of the company, but they're the commercial leaf cutter nests that are layers that you can take apart. You can, extract the cocoons, you can clean them, you can do things like that, but these are available at different stores, and the whole diameter of these are 5/16. So we compared what colonizes that nest to what colonizes this homemade nest here. We found that the layered nest, the commercial nests, they mostly attracted wasps and bees, or wasps and flies, some spiders, and the drilled nests attracted mostly bees, wasps, some flies, but there was a difference in what these two different types of traps collected. There was also a surprise to us that the most common bee was this genus species here, Ashmediella bucconus, pictured in the top left here. This was the most abundant bee, and it colonized the smaller diameter hole. So that was surprising to us that there was more colonization of that sized hole, and that this genus and this species was the most abundant leafcutter bee in the area. This table here shows the differences between the drilled and the layered nests. So here's the layered nest. This is the commercial nest. We think there's a difference in the percentage of non-bee individuals or non-bee species colonizing that nest because of the box you have to build to put around the layers that you can take apart. Because there's some gaps in that, that's conducive for spiders, and flies tend to like that, and it creates some microhabitat conditions in there that we think are more conducive to these non-bee species. But overall, we found more individuals in the drilled nest versus the layered nests, and we certainly found the percentage of bees within these nests different. So there were 76.4% of the total number of individuals within the drilled homemade nest were bees versus 22% within the layered nests. So we found quite a bit of differences simply in the communities based on the trap type. And we certainly found that if using different sized holes, you're going to census a broader biodiversity of the leafcutter bees and other bees that are occurring in the area. So this slide shows some of these bees. So the top right here, this is Ashmediella. These are all the leafcutters in Osmia and Megachile and Lithargopsis, and the subgenera. So the top right here, Ashmediella bucconus, that was the most abundant bee, and you can see it's about half the size of some of these Osmia or some of these Megachile. So that could likely be, one of the most abundant leafcutter species here in the high plains. Down at the bottom here, these are some of the parasites that we found in the nest. So Coelioxys, some of the bombyliids, some of the beetles, chalcids, and then some of the cuckoo wasps over here, Chrysis. So, within these nests, and this is known that these trap nests for cavity nesting bees, they're, they can be reservoirs for parasites and non-bee taxa just because of these microhabitat conditions and because it's nesting structure even for some of the insects that might not be beneficial.
Hey, Scott, we have a really good question.
Okay.
And one of them is, would the type of wood used to build the hives have any effect on the type of bees or the nesting results?
Perhaps so. We certainly didn't look at that, and the 4x4 posts, those were just untreated. They weren't cedar, but they were just whatever the standard 4x4 post is. And we've seen leaf cutters, they'll nest in holes and bricks and houses, and I've had insects nest in, the housing of a shifter on a bicycle, things like that.
Yeah, I think that the most important part of that is the untreated wood.
Yeah, that's it. Definitely don't get treated. Cedar's kind of a known insect deterrent, so we kind of stay away from that. But these bees, they're pretty tough, and if there's a cavity that facilitates them going in and mass provisioning pollen, they're not gonna discriminate too much unless there's a chemical repellency or something like that.
Cool. And then one question to kind of follow up on that, and then I think we're good for the moment, but that question is it a good idea for home gardeners to consider putting these bee boxes in their backyards?
Yeah, it's a good idea. Just be sure you monitor it because it will promote nesting by leafcutter bees, but it will also promote colonization by some of these parasitic species. And I think anytime you put up a trap nest, that you're going to get both the leaf cutters and you're going to get some of these parasitic species. But with that in mind, you can certainly put up the habitat. You can actually order cocoons. You can put up the trap nest. It's fun to watch when they get to provisioning, when they get to bringing back clips of leaves and pollen, and they'll disappear into the hole, and they'll come out in a few minutes, and you can kind of see them work. So that's a fun aspect to it. But they are known to house some of these less desirable species. So just keep that in mind.
Awesome. Thank you.
You're welcome. So looking at trap nests, we're trying to figure out, why the communities differ simply based on the hole sizes of the trap. So that was one of the projects we did. We found, if we would have conducted the study and said, okay, we're going to put out trap nests and bought the commercial nests, and they all had 5/16 diameter holes, we wouldn't have seen Ashmondiella bucconus that much, and we wouldn't have determined that it was probably the most abundant leaf cutter. And so now, if we were to make trap nests, then we would certainly include that 5/32 hole size, because that was the one most colonized by this leafcutter species. So once we kind of understood a little bit more about that, we set out on another project here, conducted or led by Eric Peterson. So with this study, we used reed nest bundles. So reeds are, it's like the bamboo. We cut them between the nodes, and we wrap the bundles up, and we have all different size diameter holes. And those can be used just like the nest blocks for colonization by these cavity nesting species. So this was a question using these trap nests as monitoring tools to see if these trap nests were serving as reservoirs for agrochemicals. And previous work that we've conducted has shown that around feed yards, around cattle feed yards, some of the chemicals used on those feed yards will sort of leach out into the environment. We published a paper showing they were exposing wildflower resources. So wildflowers were receiving some of these chemicals that were being blown off these feed yards. So we thought, okay, in these areas of high chemical use, do chemicals occur in these trap nests? So these are some of the things that we found. 93% of individuals occupying reeds were These first two notes are just on the community differences among feed yard locations and then reference locations, which we defined as several kilometers from the feed yard. So we had one proximal to feed yard and then one that was distal from the feed yards, but inherently was surrounded by cotton and other crops and things that the bees would forage in. So we found a striking result. 93% of individuals occupying the reed nests were wasps at feedlot sites. So of all the total number of individuals, 93% were wasps. While at the reference sites, 71% of individuals at non-feed yard sites were leafcutter bees. We don't know why there's more wasps at the feedlot sites, but that's what occurred. All substrates, mud, leaves, pollen, brood, contained agrochemicals at both sites. Mud was the substrate most contaminated with 22 of 24 chemicals analyzed, were contained in mud samples. 70% of leaf samples contained at least one agrochemical. Moxidectin was most frequently detected at feed yards, while imidacloprid was most frequently detected at reference locations. Feed yard samples contained 18 different agrochemicals, while reference locations had 10 different agrochemicals, and the macrocyclic lactones were only found at the feed yard sites, were only found in substrate at those feed yard sites. So, you know, what we found here was a little bit concerning, and it shows that as bees forage across these landscapes, you know, they collect leaf material that might have some residual agrochemicals. They might collect some mud from some wetted soil that might be laden with some of these chemicals. In any case, those chemicals are making it back to these leafcutter nests. How does that affect the populations, the reproduction, fitness, et cetera, of these bees? We simply don't know that yet. But that's some questions that are will lie ahead with this body of research. This table here shows what was on the previous slide. So the table at top, feed yard and reference. So the number of samples in the agrochemical frequency. In the first column is the matrix. So within each of those brood chambers is a capped leaf, a dead larva, feces, a live larva, leaf capsule, mud, parasites, or pollen. All of those substrates were analyzed. And you can see that the agrochemical frequency is a little bit higher in the feed yard, although even at the reference location, we were finding lots of chemicals. So, you know, this shows that these trap nests could be useful for monitoring these conditions, could be useful for, you know, further assessing how these ambient conditions might be affecting pollinator communities in the region. The bottom NMS, this is an ordination of sites based on similarities in the biological community. So this simply shows that the feed yard sites were different than the reference sites having more leafcutter bees, and the feed yard sites having a greater abundance of wasps and flies and these non-bee species. So future research aims involving pollinators. Again, this question about how specialized bees are influenced by environmental stressors based on their traits, based on what resources they use, the spatial proximity of that resource to an affected habitat, and then whether individuals like honeybees are foraging generally in both habitats and therefore potentially translocating some of these agrochemicals. So understanding traits, resource use, and how land cover influences exposure of native bees and their habitats to environmental stressors is a big area of research moving forward. Native bee specialization in agricultural landscapes, the effects on individuals and populations. So in the case of Nama hispidum, We know we can go over in New Mexico and Arizona, and this plant, you know, one of the common names is purple mat because it forms this extensive purple color across the landscape with just thousands and millions of individual flowers. Over in this area where there might be a little more landscape and habitat fragmentation, how has that affected the population conditions of NAMA, how it germinates? Has things like tilling, has that promoted more localized populations of this plant, or has it been more antagonistic? And then how are the bees affected based on those resource conditions and the surrounding landscape and farming practices? And ecological restoration to enhance resources for pollinators. That's some ongoing work. We still put that seed drill and tractor to use every time we can, when we can interact with landowners. We certainly do that as much as we can. It's a good resource to have, and it's good outreach to work with these farmers to you know, to help promote pollinator habitat. Okay, several articles here again. This Auerbach paper, the Big Gosh paper, Disqua. These are the main bee data sets from what we've gathered. So if you're interested in, and there's likely more, and some of the speakers in this series might have some additional information on the bee biodiversity in this region, but we've mined the museum records fairly well, and also have done this sort of meta-analysis across these different regional papers to kind of get a good baseline biodiversity data set for bees in the region. So, you know, now we kind of know what the biodiversity is. We're still going to pursue some projects looking at how habitat might influence bees, but we're really going to couch those questions into these environmental stressors and what are the interactions there of resource conditions, habitat fragmentation, and then bee traits, and then how does that affect biodiversity and populations. And we feel like we have, with Scotostoma prati and Nama hispidum, we have a very good specialized system there that we can package up. You know, on one hand, we can look at all the conditions related to the bees and the plants in that system. And then on the other hand, we have, you know, like honeybees that are more generalists that we know are going to visit more resources. So we can form hypotheses around both those extreme cases of resource use. Okay, that's all I have. I appreciate it again for the invite, and I think if we have any questions, I could tackle some of those.
Awesome. Yeah, we've asked a lot of the questions throughout, but we do have a few additional ones that I was going to ask. So thank you so much for giving this presentation. And if we can, we're going to put a link to a short survey in the chat. And if you are able to take that survey, that would be really helpful for us to know for programming. And also there is going to be one of the questions that will ask for your address if you would like for us to send you a seed packet of seeds that are pollinator friendly, as well as some of our guide booklets that we're mailing out to folks but at the end of this series. So if you're interested, please check out that link that Miranda just posted. And Scott, I do have a couple additional questions for you. So okay, let me go ahead and ask. So the first one that we had was when you were talking about the, oh, sorry, more came in while I was giving my spiel, so I'm going back up. Did you notice that there were any differences with the organic versus the non-organic sites that you mentioned?
No, we didn't see any effects of organic. And again, we had poor replication in our study design. I would love to ask that question, regarding farm type. But I think you would need, well, obviously you'd need to replicate, you'd have to have a certain number of organic versus certain number of non-organic within a specific crop. So on one hand, that sounds like that's easy to do, and I think it would be in some instances, but these systems are, they sit in different landscapes, whether they're close to a canyon or close to a playa. We think that the high number of chemicals in the mud substrate was in part because those bees were going to those playa margins that occurred on the feedlots. So that's probably might have been where they picked up some of those types of chemicals. But we also think there might be some chemical accumulation from wasp foraging on caterpillars and crops that are exposed, bringing those back and provisioning them.
Cool. Can you explain again what the difference between the feed yard and the reference yards were?
Yeah, the feed yards, we were just close. Basically, we were proximal within a kilometer of the feed yard, or it was closer than a kilometer. I think it might have been half a kilometer. We were pretty close to the feed lot. The reference location was several kilometers from that, distally from the feed yard. So not within several kilometers. Otherwise, outside the foraging distance, within that feed
Cool.
yard location.
Are farmers around Lubbock at all interested in reducing their agrochemical use? Or have you seen that from your research?
I think overall, I think they're interested in, they're certainly, I think any farm would be interested in reducing inputs. They rely on these chemicals because there are serious pests, that can cause some economic damage. And there's some plant technologies that might assist with reducing chemical inputs into these systems, but the seed treatment is a little out of hand, in my opinion. Every seed that goes into the ground is coated. That kind of goes against the grain of IPM in a way, because you're prophylactically treating everything. There's no tolerance there. Everything is being treated. So, and all those neonicotinoids, which are highly toxic, go directly into the soil. What's the legacy there in terms of toxicity and residuals in the soil? All the bees are nesting in the soil, or, most of the native bees are nesting in the soil. So there's some pretty serious implications there, regarding insecticide use. So, I think that the farmers would be accepting, certainly, to reducing inputs in the system. It's just their options are sometimes not that great, and sometimes pests pose a pretty serious economic problem. So it's a little bit of a conundrum. And then when you get farmers that grow things like pumpkins, that all these native bees, they're what's pollinating the pumpkins, and these are free ecosystem services, then that's kind of a deal there where, you're worrying about your inputs. You have inputs of agrochemicals, but then you also have another input, and that's a biological agent to move pollen, right? And so if you're hurting those, then you're actually might be hurting your bottom line there. So it's a tough issue. That's a good question.
A couple things that came in were, would you be willing to share your contact information for additional questions? And the second question that came in was, could you share some of the links with me and Miranda? And when we send out the follow-up email, could we share some of the papers that you mentioned and get some of the links from your references for that?
Oh, certainly. Yeah.
Awesome.
Yeah, my email address is scott.longing@ttu.edu. You can email me if you want any of these references or want to ask questions or discuss anything. I'd certainly be happy to do that.
Awesome. Did you find, if you're okay, I'll just keep asking questions.
Yeah, I'm good.
We will have to call it at 4:30, but
Okay.
did you find fewer agrochemicals in the organic fields? And did you find fewer agrochemicals in the farmers market gardens if you tested for that?
No, we didn't test for agrochemicals. So the only agrochemical testing we've done was from the trap nests, the cavity nesting bees, and the ambient collection. So that year when we collected those 300 bees, we collected those during planting. So there's some papers that show that exposure to neonicotinoids might occur when they plant because of the dust from the planting, and there's a lot of seed spillover that contaminates soil and a lot of leftover seed and whatnot. So yeah, we didn't look at agrochemical exposure in those systems. We just did those two studies, the ambient collected bees and then the trap nests.
Awesome. Are all types of bees equally good pollinators?
Well, that's a good question. And I think that we have to separate good in terms of crop production. So where we know pollinators contribute, like squash bees, and alfalfa leafcutter bees and things like that. And then, of course, all the other bees, they forage for pollen. If they're specialists, they certainly are. I would consider them good pollinators because that plant relies on them for providing that outcross pollination. For the generalists, a honeybee is a very good pollinator, but in terms of native biodiversity, are there any negative impacts of having honeybees, which is mostly a non-native insect here, right? What are the consequences of that, having big colonies, near a resource? Yeah, so I guess it's kind of a perspective there. And, if you're a farmer, you might have one answer. And if you're a conservationist, you might have another. You might say, well, they're all good pollinators because they all perform that activity.
Very true. Do farmers actively want to encourage more bees?
Excuse me, what was the last part of that?
Oh, do farmers actively want to encourage more bees in their landscapes?
I was actually, during this project, I was pleasantly surprised. All the farmers were, farmers generally understand the big environmental concerns here in the region, which is mainly irrigation. So they know that the systems are fragile. Most of them do understand that pollinators are good. So most of them do want to see bees, and most of them were, pretty excited when we would pull up with the seed drill and, drill a pivot corner or, another area. Yeah, I think they like to see bees, and the more we can educate, farmers on, which bees are maybe most effective or which crops, could benefit from bees, the better we are. There was a paper a couple of years ago out of the University of Texas that showed even in cotton, if you have a high abundance and richness of native bees, then I think there was an 18% increase in seed cotton weight just from having wild land next to the cotton farm, and that wild land, supporting native bees.
Awesome. I think there were two more questions. Were there other types of pollinators than bees in your studies? Did you see other types of pollinators?
Yeah, we saw, there were certainly other pollinators. So beetles and flies and butterflies and moths. We saw a lot of those. We focused on the bees just because there wasn't a whole lot known about bee biodiversity in the region until some of these newer studies took place. So we focused on the bees, but we certainly did see other pollinators in those other groups, flies and beetles and butterflies. We saw those. The bee bowls that I mentioned, those are biased towards bees, and they're especially biased towards the little sweat bees. So, they're inherently going to collect more bees anyway as a sampling device. But yeah, there were a lot of other pollinators out there too that are likely equally as important as bees. Bees were just the group that we focused on, that we felt like we needed to really build a background on.
No, that makes complete sense. I love this kind of stuff. This is so cool and interesting to learn about. The last question that we have for today, and guys, if there's any additional questions, feel free to shoot us or Scott an email and we can help relay some of that over. But are pollinator species affected in the same way by different agrochemicals? So are some affected more or less?
Yeah, that's a good question. They are generally affected. Well, it depends on the type of insecticide. Some of the nervous poisons are going to affect the insects about all in the same way. A pyrethroid knockdown insecticide is going to kind of affect the nervous systems of insects all the same way. Some of the more specialized agrochemicals, some of the insect growth regulators, those are going to have different effects across insect species. Yeah, that's a good question. We don't really know. We don't, in some cases, we don't know, especially for some of these specialized bees. We don't know how this exposure to agrochemicals might influence them. And really, I don't conduct this research to say, okay, agrochemicals, bees are being exposed to agrochemicals. It's likely that's occurring anyway, just because of the broad use. And it's not to point the fingers at our particular entity about, agrochemical exposure, but the more we learn about how, these species traits and habitat conditions and whether the bee's specialized or not, I mean, we might be able to say, okay, we need 2 football fields size habitat patch to support populations of Namahispidum, and having that buffer is going to ensure that Pseudoxoma prati is not exposed as it would be next to a cotton field, right? So understanding how all these interactive parts manifest to affect bees, that's where we can really understand. So maybe there's a way to build a buffer around the feedlot and to use these trap nests as, okay, we planted some buffer that we know bees won't forage on. So let's just let that habitat be, and let's try to pull the bees away from there. So, all of these different layers, they're certainly to build on success, build on as we go there, but it's certainly to find solutions to the problem, not just to document the problem.
Awesome. Well, thank you so much, Scott. This has been a super informative talk. We really appreciate your time and your being here. Thank you, everyone. If you get a chance, please go ahead and take our survey just to get some feedback on what you guys thought about today in this series. And don't forget to tune in next Tuesday when we're going to have Dr. Lauren DiCarlo from Westerfield State University talking about pollinator habitat restoration in agricultural settings. So it should be a really good build off from what the background that you've given us, Scott.
Good.
Awesome. So thank you all so much for being here. We really appreciate it, and we look forward to seeing you next week.
Thank you for having me.